So, sorry about that. So from where I was excited to introduce both speakers today. First speaker, Maryellen Geiger, and Pritzker, Distinguished Professor of Radiology at the University of Chicago. She's going to present the prize-winning project on medical imaging and data resource center. The second speaker, Andreia Vaconcellos Faria, Associate Professor of Radiology at Johns Hopkins School of Medicine, with StrokeFAIR, another prize-winning project. Both projects really go to the core of how we can reuse an important resource like radiological images to train AI model, which is a really emerging and powerful new venue. So that's my introduction. So Ellen, please go ahead.
You need to disable, because we got bumped off and got back on, you need to share the screen again. Thank you. And thank you for that introduction. And I'm very grateful for this DataWorks prize. A lot of people working here should work. Okay, great. Thank you. So today I'm going to be speaking on MIDRC, the Medical Imaging and Data Resource Center, on shareable, curated, diverse medical images at scale. We are very grateful from our funding from the National Institute of Biomedical Imaging and Bioengineering, NIBIB, as well as more recently from ARPA-H and NAIR. So why MIDRC? Well, we know that good data is the new oil. Roughly 80% of AI development requires good data that are curated, diverse, representative, reusable. So how do we do this for medical imaging AI development at scale? And that's what MIDRC is. It's a huge collaborative team science project. As I mentioned, it was initially funded by NIBIB and continues to be along with ARPA-H and NAIR. It is handled through the University of Chicago, but it is led by three major organizations: the APM, American Association of Physicists in Medicine, ACR, American College of Radiology, and RSNA, the Radiological Society of North America. It is managed through the University of Chicago and hosted on the Gen 3 pipeline. We have 7 PIs, 24 plus institutions, and over 100 investigators working on this. It's been developed over the years, established in August of 2020 with the goal, and I'll show that soon, but to do so, we have a huge infrastructure of people constantly working together, collaborating across these various societies and institutions, working on how do we develop the infrastructure? What are the SOPs? What is the data model? What are some policies for contributing and using it? How do we develop the user portal, as well as various scientific working groups and multiple research projects? So this was built for COVID as the use case. However, now with ARPA-H, we're pivoting to cancer and other diseases and conditions. So MIDRC had 2 scientific aims. One is to create an open, scalable, curated discovery data commons. That's at data.midric.org. To date, since August 2020, we have ingested over 500,000 imaging studies. These are not images, these are not slices, these are imaging studies that can include multiple images. And we've released to the public over 177,000 imaging studies. Our data is roughly, the public is around 14 terabytes, and it's coming from 22 states in the MIDRC pipeline. And our map of investigators is shown on the right. This is data that's coming from over 70,000 patients. We'll talk more about that. The other main goal is to create some machine intelligent computational capabilities, and we've done that through various open tools, algorithms, and other projects. So how do we do this? Well, from various hospitals, medical centers around the US, we collect data. Started off with chest radiographs and lung CTs. We're now moving with MRIs and ultrasound. We have 2 input portals through RSNA or ACR. The data is initially de-identified on site and then comes in where it's it's verified again by I. It undergoes mapping, bias assessment, diversity assessment, and it is hosted on the Gen3 Data Commons platform. This serves as the place for both storage, archiving, and also the user portal. Roughly 80% of the data we collect is made public. This is AI ML ready, fair, trustworthy, representative, and all researchers around the world have access to it. To enhance the data, we index and interoperate with other data commons. For example, we interoperate with biomedical through the Biomedical Research Hub with the Biodata Catalyst, as well as N3C. Because we are medical images, we collect medical imaging, we need to interoperate to enable multimodal research. What happens to the other 20%? We sequester that by patient. This is not publicly available. We match the distribution of that data to that in the public, and we use this, as I'll note later, for translation through regulatory. So how do we do this? Well, we first get started by working with different hospitals and institutions and work through them with the data transfer agreement. We have a lot of experience with ingestion, management, and curation of DICOM imaging data and related clinical data from various sites. And so we work with the sites to do the technical setup while they're conducting the legal and regulatory steps. As I noticed, we have two pathways into MIDRC, either through ACR or RSNA. These sites involve de-identification. On Gen 3, all data is fully de-identified so it could be shared in an open way. We also undergo data harmonization with the DICOM images and clinical data, which is submitted in various formats. For example, our MIDRC data dictionary, which you can find on the website, is necessary to organize the data and their attributes in a relational model. We are constantly increasing this data model as we take on different and newer cases, both modalities and disease conditions. We also have the data undergo harmonizations. We've developed a link mapping protocol, which allows us to take, for a given imaging type, there's multiple names out there depending on what institution you acquire the images from. But that makes it difficult for data cohort building and searching. So with the link mapping, we are able to relate those to a common name to enable the linkage. So we've evaluated these methods. This one is over 97% of the studies were able to be matched to 65 unique link terms from 1,000. And it's regularly updated as we increase our data types. And all of this is public on our MIDRC GitHub, where you can pull down and use this mapping with your research in your lab if you like. We also intake annotations and labels, both from within MIDRC as well as third-party sources. And we've also have a user platform on the Gen 3. This supports the DICOM-based images we take in and allows for searches based on the images and the metadata. We also we integrated a browser-based OHIP DICOM viewer so one can view the images as you're looking at them. Diversity of the MIDRC data is extremely important to us. It needs to be representative of the population so that folks can develop ethical and trustworthy machine learning algorithms. So based on the metadata, we assess the diversity of the data, and we continue to do this over time. Currently, our contributions are coming from 22 states. Here's an example of race and ethnicity over time. And what we do is we compare our data to the US Census and the CDC COVID positive cases. Here it's being compared using the Jensen-Shannon distance, where 1 indicates there's no similarity and 0 indicates very similar. And you can see here that over time, as we did the comparisons, we get very similar cases. And this is important to understand the diversity as you developed your AI algorithms. We've now included this into an online diversity calculator, which is also on our GitHub. It is generalizable. we can compare our data sets to the different to CDC or US Census or to any other data set. And this can be used beyond imaging data sets also. And so this will be part of the ARPA-H biodata medical data fabric. I've noted that we interoperate, and we've done some multimodal research using two different ways to link the cases, the images in MIDRC, with the two external data sets, Biodata Catalyst and N3C. Now, I noted that the 20% we put on the side for that is sequestered and no one has access to this, and this is to accelerate translation of it. With the code for doing the sequestering is on our public MIDRC GitHub. It can handle multiple attributes, how you would like to split your data, say into training and testing. For our 20%, we will be using this with companies to help them go through independent testing and regulatory. For example, when an investigator submits their request saying what is their claim, what is their intended population, we will draw from our sequestered set that distribution and then draw cases that will be used in testing. We will report the performance, and we conduct this in a secure, containerized area of MIDRC, and we will then work with them to report it to FDA. Ultimately, we'll also be incorporating our diversity and bias tools into here. This is being developed with members from MIDRC, industry, and FDA, and it's our route to sustainability. I mentioned our GitHub. We have various tools and resources. You can go to our website under algorithm. You see many listed with links to relevant publications as well as the GitHub. One example is our metrology decision tree, which is developed for having an interactive decision tree to assist MIDRC users in selecting metrics and reporting performances. And you can walk this through, and we've now converted it to for use with various oncology analyses tests. We also have a bias awareness tool, so investigators can learn about the different biases that may occur in the development, testing, or use of an AI algorithm, and giving them methods on how to mitigate such bias. We host grand challenges, and we're currently in our third one on decoding AI decisions for pneumonia and chest radiographs. And here, the winner's code is always then published on our MIDRC public GitHub. We continue to engage with the national AI ecosystem through our funding now with ARPA-H and the Biomedical Data Fabric to enhance the scalability of the national infrastructure for AI. We are one of the phase 1 BDF performers here. They've asked us to switch from COVID to oncology, and we're doing that. We're taking our developments in MIDRC from our initial funding and creating tools for others to use. We're also expanding MIDRC to index across other imaging commons, such as here, we can index MIDRC along with imaging data commons and the TCIA data. We're also working on extending our interoperability and obtaining data from other sites using EdgeNode technology so that we can bring it into MIDRC both in a centralized and distributed data format to enable further multimodal developments. For example, this is on our new MIDRC BDF imaging hub, where we enable indexing, where one may be searching for a specific image cohort, and here you can see that it will tell you where these images may be, not necessarily in MIDRC, but in another one. Our harmonizing, indexing, and interoperability continues both with our public data set, as well as investigating how do we handle restricted data and to allow potential investigators to find and use such. We're funded by NIR in their NIH pilot program, and we're also listed on the NIH website as a means for investigators to share their data with others by placing it within MIDRC in the scientific data sharing methods. We're creating community. You can always check our website to see where we are by our MIDRC by the numbers, showing how many studies we have, how many patients, how many presentations? 58 publications. We have over 800 registered users of the data site. And you can follow us with our monthly seminars. Everything is recorded. It's on our YouTube channel. Our software is on the GitHub. And of course, our data is on data.midric.org, which is open. So in summary, we can continue to grow and serve the medical imaging AI community. We want to continue our sequestered data commons and to enable more efficient, more effective translation of AI methods. We will continue to extend our assessments of bias and diversity into metrics that others can use, and continue our methods for indexing and interoperabilities. We continue many talks with partners from various funding agencies, including groups on molecular imaging, stroke, cardiac, point of care imaging, and digital path, and others. And so we continue to aim for a representative AI medical imaging data commons of all populations across the nation, both centralized and distributed. So at this point, I will stop. I want to say thank you to our funding agencies, as well as to the folks in the ODSS and the FASEB for this DataWorks prize. Thank you.
Thank you so much for this overview and for sharing the details of the project. We're going to have question for both presenters at the end. So I'll invite Andreia to join us and present the work of the project. Thank you.
Thank you. I'm going to try to share my screen now. Can you see my full screen?
Yes.
Okay, thank you. So my name is Andreia Faria. I'm Associate Professor of Radiology at Johns Hopkins, and I will present our work on the StrokeFAIR, which is a public dataset and analytical tools for brains of patients with stroke. First, thank you very much for the invitation today. And overall, thank you for the prize that I hope will allow us to disseminate our work and inspire other people involved in data sharing. So our team involves radiology and the neurologists in the clinical side, and also the librarians of our institution that were very helpful on guiding us on regulatory issues and how to share our data and how to do it according to the principles of that regulate protected clinical data and the other source of medical data. So our work focus on patients with acute stroke, as the motivation for this work was actually related to clinical research and clinical questions. On one side, we know that stroke is the source of most of our knowledge for the last 100 or 200 years about how the brain works. So it's a model very, very powerful to understand location of functions in the brain. You knock out a region, you have a functional consequence. And as much as these lesion-based studies evolved over time, we still have a lot to learn with them. And on the other hand, it's also, of course, quantifying stroke images is very important in clinical decisions as metrics of volume, of stroke core or hypoperfusion, or quantification of regions affected can guide clinical decisions and our prognostic models, create prognostic models that can be of clinical relevance, both for acute medical care and research. But how can we do that? These strokes are very variable, right? They're very heterogeneous, and the populations are super heterogeneous, so we could do something like that. only if we accumulate large amounts of data that represent not only the variability of the disease itself, but the variability of the population. So we need knowledge base that are appropriated to feed our models and to analyze these data sets. Of course, it would require technical developments in terms of automation. So this is our overview, the overview of this project. So we first needed this knowledge base, and then we would have to create automated models because we were working with big data, to determine where the injury was, segment this injury. We need information about locations on the brain to quantify locations affected. And then we would need to output this information in a way that it would be understandable to people working in different fields, from computational scientists to clinical scientists, because then they could we could really create an open source and the open science source where people not only have access to the data, but they have access to these objects that they can easily understand, work with. Therefore, coming to our initial aim of creating models and creating means to help people with stroke, both on the clinical side, creating like subgroup patients in subgroups of clinical relevance, or creating more resource to understand brain function after stroke. So first step was to create this knowledge base. And of course, I will not repeat the FAIR principles, but I think the data, the days that we could refer data available upon request in our papers or things along this line are finally gone. And I think it's nobody question anymore the power of having your data not only free, but also organize it in a way that people can understand and use it. So we would need fair, findable, accessible, interoperable, and reusable data. And at the same way, we have the same tendency for creation of tools to analyze this data. So we also focus on development and use of a FAIR research software. When we started this project, we thought we didn't have to initiate these initiatives because we hoped to find lots of data and lots of software to work with available online. And it turned out that that wasn't the case. So we had to initiate this initiative of creating FAIR data for acute stroke patients. And what we did was to collaborate with our stroke center and transfer data from our stroke center under hospital firewall for anonymization, for annotation of the stroke area. And then we created, we de-identified data, we went over all the steps for anonymization is, as people say, is never always possible in a way that people will never re-identify later on, but we did all that was possible given the bioethical regulations. And we organized also metadata, and for stroke, this is a little more straightforward, I would say, because of the American Heart Association Get With Guidelines initiative, where many hospitals, hundreds of hospitals, actually thousands of hospitals are enrolled in this initiative. And it's a national database where people with stroke, hospitals register people admitted with people with stroke. So we had this data this metadata already standardized in some quantification parameters of clinical tests, laboratorial tests in the admission. So for each patient, then we have basic clinical data plus images anonymized under the firewall. And then we moved this to data repository, professional data repository located in Michigan, ICPSR, that handles things that would be hard for us to handle, like data usage agreements and all the paperwork related to data download and maintenance. And this alleviated a lot of our work on data management. So having this now both on our side and the public, we were able to go to the next step of creating tools, automated tools for reuse and analyze this data. But first, I want just go over the structure of the data. So we now have about 3,000 subjects with multiple different kinds of image at ICPSR, and the images are raw images, minimally processed, post-processed images with strokes annotated, and a lot of metadata, a lot of different imaging formats that one can access and download from the ICPSR address that I'm going to show in a minute. We also have all sorts of stroke frequency maps in different templates. And yeah, this is the address for data download. And the data actually become the first successful delivery was about a year ago. And we have created a good number of downloads right now. And this data management plan, actually, how we manage the distribution, the organization and distribution of data, actually valued us the data management plan prize last year. So now we had the data, we started to develop AI and machine learning general algorithms to go over all the pre-processing, data segmentation, lesion segmentation automatically. And we did that through a combination of classical machine learning methods and deep learning. And we actually were able to create it using about 2,000 cases and method for segmentation of acute ischemic stroke that, of course, we also made public. I'm going to show in a minute the repository to download all of this. And it's quite simple to use. And I mean, it's simple for computational scientists and simple for clinicians, clinical scientists that can just upload an image and download the results. And together, we now knowing where the lesions were, we had to develop means to attribute labels, location, spatial labels to these lesions. And we did that by creating brain atlases that give us the information about locations in the brain. Some of these atlas, again, I think it's a great tip of Dr. Warner in the beginning of this seminar, is that don't reinvent the wheel, and we did not. So we used a lot of resources that were available there for the identification of the data organization, many atlas that were already available, some of our groups, others not. But we also developed new tools where we needed them. So we developed this digital atlas of arterial territories and other atlas that I'll show later, using the frequency maps of our thousands of patients with stroke. And again, we also made this all available through MIDRC, which is the neuroimaging tool and resource collaboratory platform. And By doing that, people can download this deformable digital atlas that corresponds to different criteria for segmentation and for localization. Some of them more general about classical anatomical structures, others more focused on arterial distribution or indices that are important for stroke. And we also release atlas and metrics for people being able to do quality control. So to see how their mapping is satisfactory compared to the template, and therefore how good their localization method is. Okay, now we had the injury, the location, and we had means to calculate simple indices of how many brain structures were affected by the stroke. So we warped all these methods in a tool called ADS, Acute Detection and Segmentation Tool for Acute Stroke. And this is also open source, available on MIDRC and also GitHub. And what this tool does is given a MRI of DWI and of a patient with brain stroke, it quantifies it segments the stroke, output results, output percentage of lesion loadings of regions affected by stroke, and also some important indices for acute stroke that I'm going to show. So we are therefore pretty close to our initial aim of not only develop models that are important for stroke, but allow people apply their own interests to develop such models in a public data set using public resources. And that's what we did. Now we have this open platform where you submit your image, you get proportion of regions affected by the stroke. From there, you are able to train models. And here I'm going to show just one example of a model we trained for transforming these lesion loadings in words. So we pretty much were able to generate automated radiological reports that agree with the radiological reports. And one thing that's very important in development of any kind of machine learning or AI technology is to expose features that were implemented in these models. So we not only create these models, but we make sure that each of these models output what the model is using to such classification, so people can check if that makes sense, if the model is using something that's biologically relevant. So these models become much more effective and much more likely to be adopted in the research and medical community. So here is just one example of one of these applications. So you submit an image, you get these automated reports, you get your mask of strokes and regions affected, and you can use these to create such kind of models, or you can develop your own, you can use both the raw image and the processed image to test, for instance, your own hypothesis, or to aggregate data and develop your own research on any kind of interest you have. By doing that, we were able to initially explore this data set, looking at associations between function, clinical markers, images, and studying, for instance, mostly on language. So we studied, for instance, a lot of relationships between lesion and language in these patients. But that's, I guess, not what's more important. And here, what I think is more important. So this is from last year. This is, we stopped at, actually the graphic stopped in December last year. And here is how people are using our data set, our automated reports for our automated methods for stroke quantification, our atlases of vascular territories that actually got us to this DataWorks prize. And also, up to December last year, this is how our resources have been used. The map actually corresponds to the data set, how many universities and in different countries are using it right now. I believe the list now is growing, and that's our aim, of course. We would be naive and arrogant if we think that we could explore everything that the richness of clinical data would give us. So I think our own contribution is always in different scales and in different amplitudes, share raw data, share knowledge, and share resources, and allow really open science to launch. So I guess that's it. Thank you for inviting me, and thank you for the prize, of course.
Fantastic. Thank you, both presenters. We have some time for questions. I know we already have one question in the chat, but I want to start it with one thing that, it's pretty striking. You both are, really focused on radiology. You're both radiologists, right, as a modality. Well, your radiology sort of like as a particular area. What is special about this community compared to other areas? Because imaging is prevalent across biology, but what is it specific about this community, in your opinion, that really allowed these two projects to flourish and be successful? I don't know if that...
Well, I could start. Well, first, for AI has been being developed for medical imaging for decades. So we've had a head start there. Also, medical images are extremely organized around the world. One can get a data set from anywhere, and it will be from any, say, CT scanner, chest radiograph, whatever. It will be a DICOM image. So they already have very standardized means of acquiring the data, so that the parameters of the acquisition are included in this DICOM header on the DICOM image, which makes it accessible. The other thing is, like you said, there are many images around. Often, I think people have not looked into what's in the image to relate to disease as much as they could because of the availability. And so that is why I think the need was to acquire these. Plus, with these newer deep learning methods, you need a lot of images. And you need to not just have images, but you need to understand where they came from, how are they curated, how are they harmonized, so that one can do appropriate deep learning developments, as well as knowing the diversity of it to avoid bias. I'll stop there.
Yes, yeah, I agree. And I think, yeah, one point in our favor, I think, is that, yeah, deep learning, AI, not only deep learning, AI initiatives in general, machine learning, etc., initiated not with magical images, but utilizing a lot of images that so they know very well. So the method is mature in this area. And yeah, I completely agree. I can't agree more that we produce tons of images every day in hospitals. And we have this experience on working a lot with research images. But I think clinical images are still very underutilized. So we do these scans, these X-rays, these photos, and each clinician looks at it qualitatively, take conclusions, that's it. So if we are able to develop a pipeline where people can utilize the richness of this, all these or some of these clinical images that we have every day, and people look at it in this regard, if we can put them back in the research or in the biomedical engineering pipelines, I think that will be a real revolution in terms for radiology and health care in general.
And I think it's really nice to see those two projects together, because I think you really need to close the loop, to your point, to demonstrate the utility, to demonstrate what you can do, to demonstrate that you gain novel knowledge by applying AI to those resources to justify the accumulation of resources. So in your opinion, what is the next area where we should be focusing on? What is our hope, your hope personally?
I can start it. Yeah, I think one of the greatest difficulties we have, and we still have, is not from the technical side. The technical side, I would say, is easy. The greatest difficulty is actually on the regulatory side, and it's a big deal to try. I think all the presentations up to in this price up to now had the same mode, and I believe the others that are coming will have the same mode. I think one of the greatest challenges is to develop this standardization of language, standardization of approaches. And I completely agree with Dr. Werner said in the beginning that consensus is different of majority decision, and certain points decision have to be arbitrary. But the greatest challenge, I think, right now is to get harmonization, to get comprehensive data. If we are able to overcome this with the minimum cost, so people can easily aggregate their data, either in a centralized approach or in a decentralized approach, we will make use of all these decentralized resources that do not go to the big data set, or do not go to contribute to this knowledge that you only find on heterogeneous and large amounts of data. So I think to overcome regulatory issues for data aggregation and develop tools, public, comprehensive, accessible, for overcoming this issue, like the identification, anonymization, aggregation tools to make data computable objects so you can easily aggregate them, then we would be able to make use of all this knowledge that we just throw out day after day.
Maryellen, you're on mute.
Sorry. Yes, I agree. We have the technology, but it's the collaborative, people working together, collaborative, different data commons. And the problem is with different data commons, they all have different governances. And so while we're very open, other data commons are not. And to really advance the research is to first make sure the medical images are highly harmonized and curated. I'm not saying high quality imaging, but curated. So we know these are good images, these are not so good, because that's the real world data. So one is to do that, but then also to enable interoperability with non-imaging commons, those that might have more EHR data or genomics data. And with every commons having different governance, it's difficult for a user to have access to everything. And it's that working together. Within all these commons, people seem to be working very well together. We have 3 societies working very well together to move MIDRC along. All different expertise are involved in moving MIDRC along. However, then all interoperating, it requires means to link. And while we have the technology to link the different, getting over all these different governance that some are more open than others, that's what we need to focus on.
And I think
also,
go ahead.
Also, you asked another one. My other one is to take our knowledge of how we've standardized and worked together in medical imaging and translate it and work with the folks in digital pathology, where they're still trying to harmonize their methods of acquisition, depending on the different acquisition systems. They not, it would be great if they were all DICOM, but they're not. So to kind of work with them, and maybe they're newer, to bring them up to the where we are and to collaborate with them. I think that share our knowledge.
Yeah, I think that's one the point of making at the beginning. That's one of the advantages of that particular community, that you had that work done over the years to enable what we have seen today. We are about at time. I know we're going on break, so I'd love to continue this conversation. But I think I thank you both presenters, and there is a question in the chat that maybe Andreia can handle quickly, and then I'll yield back to Emily. And again, thank you for both presenters, and thank you all.
Thank you.
Thank you. I don't see the question.
The question is related to ICPSR, and are data consumers paying for that, or how are the costs related to that curation managed for your project?
Yeah, I should have mentioned that. No, nobody pays. It's public. We had to pay, and which was not. It was hard for us because we had to utilize other resources. This work is not funded by NIH or anyone. It was almost a voluntary work. And so we paid for some data curation in ICPSR, but nobody has to pay. The data is public. The data is public, but because our IRB required is restricted access. Restricted access means that you have to submit you have to ask the data, but you have to have your university IRB approval of the project you were want the data for approved by your IRB. That's it. There is no other requirements. You don't have to cite any of us as authors. You have to cite and you use the data set, of course, but that's it. Having done that, you can download the